Thanks, John. It is my great honor to introduce Dr. Richard Heyer, Emeritus Professor of the University of California, Irvine, as the actually 2020 winner of the Lifetime Achievement Award. I can speak from personal experience. Rich's desire, interest, and ability to help young investigators thrive. He did so with me. I was I saw that a meeting was happening in Riverside, California, Newport Beach, that was brain imaging studies of intelligence. This was almost 20 years ago. And there were luminary speakers, including Paul Thompson, Vivek Prabhakaran, Jeremy Gray, Alyosha Neubauer. And I contacted Rich to see if this recent graduate could insinuate himself into the program, and he was excited to let me do so. And it led to several years of productive collaboration. I would add that Rich is perhaps going to talk to you about the Heyer Prize, in which an anonymous donor gave 10,000 unrestricted funds to 8 different young investigators who are studying the biological bases of intelligence, some of whom have given talks this morning. They have a little HA next to their names in the program. He has been a tireless advocate for intelligence, early participant, and a great colleague and friend. Thanks, Rich. I look forward to your talk.
Well, thank you. That was a very nice introduction. And I don't see how to share the screen here. I don't have a button to do that.
Perhaps John can talk you through it. I think it's down at the bottom.
Oh, there it is. I got it. Okay, that should do it.
There you go.
And you can, whoops, you can see my slides here. And let's do that. And I'm really happy to see some old friends here in the audience on Zoom, some new friends, some new faces. I'm glad to see you all. It's exciting for me to have this opportunity to reminisce a little bit about the last 50 years. Most of you, or maybe half of you, have not been born when I started. So I want to give you a little historical perspective. There are really 2 purposes of my talk today. To give you a historical perspective from my personal point of view, and also to encourage you to think about the big picture, however you think the big picture should be defined. My professional evolution, you can see illustrated here, age 10, performing magic at my younger brother's birthday party, to my chemistry set at about age 12, to brain imaging when I was a little older. I'm not going to go through all the steps in between, but I do want to talk a little bit about some of my first thrills at graduate school, and then at my first job at the National Institute of Mental Health. In graduate school, I was very lucky to work with Professor Julian Stanley, himself a winner of the Lifetime Achievement Award many years ago. I started Hopkins in 1971, the same year Julian Stanley started the study of mathematically and scientifically precocious youth, which I worked on. I passed out pencils at the first talent search. That's what graduate students did. And I also learned a tremendous amount about psychometrics at Hopkins, not only from Julian Stanley, but Bert Green, Warren Torgeson, Alphonse Japanis. Really was very well versed in that, which came in quite handy later. After Hopkins, my first job was in the intramural research program at the National Institute of Mental Health, where I worked with David Rosenthal. He was the director of the Laboratory of Psychology and Psychopathology. He hired me to help work on some of the personality data from the Denmark adoption studies of schizophrenia. I learned an awful lot about genetics from David, wonderful human being. And by chance, my office was next door to Monty Buxbaum, who was working on EEG and evoked potential research. And so I learned a lot about that from him. Now, for some reason, I'm frozen here. And let's see why the slideshow is not progressing. Oh, there we go. So my first job after NIMH was at Brown University, the Department of Psychiatry, where I used what I learned from Monty Buxbaum to set up my own evoked potential lab on an Apple II Plus using grass amplifiers. We recorded from 32 electrodes. And I published some work correlating evoked potentials with the ravens. And so this was the early work I did on the brain. And meanwhile, back at NIMH, I had already left, but Buxbaum had acquired one of the first PET scanners. And he used that to study the Genane quadruplets, a case that was being studied at NIMH for over 20 years. The Genanes were genetically identical quadruplets, all concordant for schizophrenia. As youngsters, they performed, they were quite famous, as quadruplets were at the time. When they got older, they all had schizophrenia. And they were among the first people at NIMH to receive PET scans. Their pseudonyms, by the way, Nora, Iris, Myra, and Hester, spell out NIMH. And Monty Buxbaum used this PET scanner to do scans, which was the first time this had been done in schizophrenia. I got to see these images before they were published. As soon as I saw these images of functional activity in the brain while people did tasks, I knew instantly that this was going to be the future of psychology, and particularly the things I was interested in. This is around 1983, by the way. Just for perspective, most cognitive psychologists began imaging with MRI around the year 2000. So this was really quite early. And the first study I did with PET, I moved to the University of California, Irvine, because Buxbaum had moved there. He acquired a PET scanner at UC Irvine, one of the first commercially available PET scanners. And I joined that group, and I was able to do a study of 8 individuals while they performed the Raven's Advanced Progressive Matrices, while the fluorodeoxyglucose was settling in those parts of the brain that were working the hardest. And the PET scan showed this remarkable result of the person with the highest Raven's score of 33 actually had lower metabolic rate than the person with the lowest score. This was counterintuitive at the time and really was the basis for what became known as the brain efficiency hypothesis of intelligence. This was so new in 1988 that it was covered by Newsweek magazine. There's a younger version of myself, How to Tell If You're Smart, See Your Brain Light Up. This kind of publicity was almost unprecedented for intelligence research. And I just want to emphasize how thrilling it was when we first saw these images. No one had seen this before. Nowadays, of course, these images are ubiquitous, and it's no big deal to see these images. This was really thrilling. This is the actual data from that paper. We had 3 brain slices because our PET scanner only did 1 slice at a time, and then the bed automatically moved in for the next slice and then the next slice. So we got 3 slices. We took the image. We had an algorithm that outlined the skull. We identified quadrants, And then we did a mathematical peel of the cortex into these sectors, left and right. And then we looked at the average glucose metabolic rate in each sector, which we then correlated to the Raven's score. So this is one slice, a second slice, the third slice. And these correlations with an N of 8, some were significant. They were all negative correlations. And they were not just in the frontal lobes. They were dispersed around the brain. Our first hint of brain efficiency and our first hint that areas distributed around the brain could be related to intelligence. Now, just for perspective, this kind of analysis, barely rudimentary by today's standards, was about 11 years before the first version of SPM, a standard analysis program, became available. And if you compare this to what we hear about today, don't laugh, because these early studies really set the research agenda for what we're seeing today. I just want to talk a little bit more about the thrills of PET scanning in those days. To get a scanner and a cyclotron, first scan cost about $3 million. The second scan was charged at about $2,500, so an N of 8 cost $20,000, and nobody was giving grants for this kind of work at that time. But UC Irvine, while they got the scanner, they didn't have a cyclotron. The closest cyclotron to make the isotope needed for PET, with a half-life of 109 minutes, was 400 miles away at UC Davis. So we had a woman in Newport Beach who had a private plane, and she volunteered to fly up to Davis. There was an airstrip right at the cyclotron, so she would taxi up there. They would load up the fluorodeoxyglucose that they made that morning. She would fly back to John Wayne Airport, which is a 10-minute drive from our campus. And if we were lucky, we would have enough isotope to do 2, maybe 3 people that day. And on days when Davis was fogged in, we had to rent a jet plane to do this. So while you may think an N of 8 is not worth thinking about, I can only tell you it was like the Wild West to get this sample. And in 1988, Arthur Jensen actually arranged for me to get private funding for a small conference of about a dozen people to discuss the impact that this kind of PET scanning would be having on intelligence research. And had many conversations with Jensen over the years about this. In 1990, I published an editorial in Intelligence talking about the end of intelligence research and what we would have learned when we're all done. And in that editorial, I basically said every psychology department needed its own PET scanner. This is 10 years before MRIs became more available. So, and now, of course, virtually every major psychology department has more than one MRI scanner. In 2003, I had a conference, I had a symposium in the the ISIR annual conference, which was here in Irvine. Rex mentioned this. There he is back there, Alyosha Neubauer. Paul Thompson came down. He was at UCLA at the time. Paul Thompson now runs one of the largest multinational consortia collecting brain imaging and DNA data. And he's still very interested in intelligence. So over the subsequent years, I had more thrills of discovery, both with PET and MRI. And I just need to note that there were no external data sets to draw from at that time. So all our early studies had ridiculously small samples, which we generated ourselves. And it's fair to say they require replication using today's large data sets and much more sophisticated analyses. But I do think these early studies did help set the research agenda as far even into today. So I think many of you are familiar with our Tetris study. This was a study of learning where we hypothesized that when you learned a complex task, your brain would become more efficient. We had normal volunteers practice Tetris every day for 50 days. This is their learning curve. We scanned them here at day 0. We scanned them at day 50. And sure enough, their brains became more efficient. By the way, this study appears in the Guinness Book of World Records in their gamer edition as the first computer game studied with brain imaging. With Camilla Benbow, we did a study that really emanated from my Hopkins days with the study of mathematically precocious kids, where we got male and female college students based on SAT scores, either over 700 on SAT math or in the 500, 550 average range. These are group average images, 11 people in each group, at the time, the largest PET scan study. And what we wanted to test to see if there was brain efficiency in males and females. What we found was in males, positive correlations between scores on an SAT math test they took during the uptake and metabolic rate in some parietal areas. In women, there were no correlations at all, basically, even though the women were perfectly matched to the men for math ability. This was a very early indication that there were sex differences in brain function that needed to be explored. I also did this little experiment, interesting story behind this, but an oil company actually paid for this study. And they wanted to know if a test of divided attention could detect people who were mildly impaired on alcohol. What we found was interesting in that half the sample, when they got alcohol, their performance on a test of divided attention increased, and their metabolic rate actually got higher. But the other half of the sample, when they ingested some alcohol, their performance actually decreased, and their metabolic rate went down. Again, more evidence of the importance of individual differences. We moved to MRI, structural MRI, around 2004, when we got some data from Rex and pooled it with our data. And we correlated the amount of gray matter using voxel-based morphometry to IQ scores. And in the full sample, we found certainly the frontal lobes were involved, but also there were these other areas, more cognitive processing areas. And then when we reanalyzed the same data by men and women separately, rather than partialing out sex, we did separate analyses. And sure enough, the men had this large area back here in the parietal lobe, pretty much the same area where Einstein's brain had more glial cells, by the way, and also the frontal lobe. But the women were mostly exclusively in the frontal lobe, Broca's area. And this was more indication of how important it would be for future studies to analyze the data in men and women separately. With Roberto Colom, who came to spend a year with us, we tried to correlate the amount of gray matter involved in subtest scores from the WAIS. This is the ranked number of significant clusters of not activity, but of correlations where gray matter was correlated with the subtest score, and the ranked G loadings of the subtests. And you can see there's a nice linear correlation here in our effort to look for a neuro G. A lot more work on this has been done by various groups. Then we tried to replicate the Tetris study with Rex in 2009, and we did both structural and functional MRI. The blue areas show where there was a functional difference before and after a group learned Tetris. The red areas show where there were some structural differences. Again, this was an underpowered study, but the interesting thing here was there was no overlap between the functional and the structural areas that were related here. Again, an early indication that there was something about the relationship between functional and structural changes that was not going to be straightforward. So the lesson from all these studies, all these early studies, is brains are like galaxies. The same basic principles, but no two work the same way. In other words, individual differences are absolutely crucial to understanding what's going on here. I also want to mention we did a little pilot study where we had individuals with low IQ for no known genetic reason, IQs of around 75, a small group of people with Down syndrome and normal controls, age-matched normal controls. And consistent with the idea that low IQ individuals would have inefficient brains, we saw a little bit of evidence for that. But we were able to use this as pilot data for the next thrill of my career, which was to get major funding from the National Institute of Child Health and Development. The grant was Cerebral Glucose Metabolism and Down Syndrome, Impact of Dementia. You may not know that middle-aged people with Down syndrome almost always get Alzheimer's disease. And so this was of great interest in terms of early detection of Alzheimer's disease. And we published a bunch of papers on this, but the big chill was, unfortunately, this grant was not renewed, which led to my retirement in 2007. But 2007 also was another thrill of kind of putting everything together up to that point. With the publication of the PFIT model with Rex in behavioral and brain sciences, along with commentaries. And this really, I think, helped define the subsequent imaging studies. And also in 2013, I created the series of video lectures, The Intelligent Brain, for the great courses. And this has really been, I think, important in help educating the public, the interested public, about what intelligence research really is and what it is not. And then the next thrill was publishing my first book in 2016, The Neuroscience of Intelligence, part of a series by Cambridge University Press. A couple of weeks ago, it's still #2 on the bestseller list at Amazon in the neuroscience category, even though it bounces around a lot in terms of its ranking. There's still a high demand for this book, and I'm very proud of it. When I was invited to give a talk at Frankfurt, Germany, a couple of years ago, they presented me with this photograph, this collage of people all around Frankfurt reading my book. And I got a lot of laughs over this, and I really appreciate this. Recently, another thrill released by Cambridge University Press, an edited volume, The Handbook of Intelligence and Cognitive Neuroscience, that I worked on with Aaron Barbet and Sharif Karama. But almost immediately, the recent chill was Cambridge canceled the second edition of Earl Hunt's book, Human Intelligence, which we were calling The Science of Human Intelligence. I wrote this with Roberto Colom. Not exactly clear why they canceled this, but they got some reviews that seemed to indicate some people might be offended by some of the material. And so we are now working to get another academic publisher. But back in the first book, I enumerated 3 laws. Nothing about the brain is simple, no one study is definitive, and it takes time to develop a weight of evidence. These are not terribly controversial, but I also asserted in the book that the ultimate goal of intelligence research was to increase intelligence, especially G. This turns out to be a bit more controversial. And if you don't like my goal, my question is, what's your goal? And why should young researchers spend their career pursuing it? So if you're working in psychometrics, what's the ultimate goal? Same with neuroimaging, genetics, cognitive processes, development of intelligence, learning, memory, education, social or cultural influences, group differences, animal intelligence. If this all doesn't explicitly or implicitly lead sooner or later to increase G, then why are you doing it? You can say, well, we want to understand something better, but understand for what purpose? This comes under my purpose of getting you to think a little bit more about the big picture. So, and if you agree with my goal of increasing G, you shouldn't be distracted by inarticulate questions like, isn't this scary? Or aren't there ethical problems? Or isn't this eugenics? Or aren't there more important things than intelligence? Can you even define intelligence? Such questions do raise important points, but they need better articulation and they should be discussed. Isn't this scary? Not to me. Why is it scary to you? Aren't there ethical problems? Tell me, what do you think the ethical issues are? Isn't this eugenics like the Nazis? Not even close, if you want to understand what eugenics is. Aren't there more important things than intelligence? Of course there are. I don't know how to increase them though. I'm working on this one because this one is important. And can you even define intelligence? Now you should read the first chapter of my book if you haven't already done so. In my opinion, the key to progress for the next generation of intelligence research is neuroscience experiments that incorporate individual differences based on brain imaging, like connectivity, and DNA, like polygenic scores, using new psychometric tools like Pathfinder to test hypotheses about causality using brain manipulations. That's what makes it experimental, like transcranial magnetic stimulation or drugs. And think about the big picture of what such experiments would reveal. Now, the two most complex things the brain does, consciousness and intelligence. Let's think about a big picture here. Are consciousness and intelligence related to each other? For example, do high IQ individuals require more or less anesthesia for surgery? I've asked a lot of anesthesiologists and they don't have any idea about this question. Do anesthetic drugs cause functional neurocircuitry changes among PFIT or other intelligence areas? Well, here's an image of a person that we studied with anesthetic drugs way back in 1995. These are hard studies to do, but we managed to do them because I was working with an anesthesiologist who was a resident at the time, who was really gung-ho about using PET scanning to examine some questions about how anesthetic drugs work. And so we brought normal volunteers into the imaging lab and scanned them while they were awake, while they got enough anesthetic drug to be barely responsive. And then when they got enough drug to be fully anesthetized, we were testing a neurocircuitry hypothesis about what anesthetic drugs do. And we subsequently published an interesting paper where we correlated the change in glucose metabolic rate during one anesthetic drug, isoflurane, with the density of muscarinic receptors in the different areas that we could look at with PET. And we found a positive correlation of 0.85, suggesting that for this particular anesthetic drug, this particular kind of receptor would be important. Similarly, we did a different anesthetic drug, propofol, with the density of diazepine receptors. and found a negative correlation of 0.86. So I want you to think about substituting a low dose of any neurotransmitter, agonist or antagonist, or transcranial magnetic stimulation, and look at changes in brain connectivity in high and low IQ subjects. Look at it by age, by sex, by polygenic scores, and of course, control for SES. And to do this, you have to collaborate. And even if you don't think you have the skills to do these kinds of experiments, you have a lot of knowledge and technical ability in psychometrics and other aspects of intelligence research that are absolutely crucial to collaborating in this kind of work. So I want you to think big. So Rex mentioned the higher prize and, that was enabled by a donor, and he's interested in the biological basis of intelligence. And ISIR awarded this prize to 8 individuals in 2021 who are working on everything from gene expression to cellular function and structure to brain networks to cognitive processing, all the way up to psychometric intelligence. Now, let's see, you've heard from Matt, you've heard from Kristen, you've heard from James. Tomorrow you'll hear from Erhan, you'll hear from Annalena, and you'll hear from Natalia. They're doing really excellent work. If you're not familiar with these people and their work, you might take some time to look at it. It's really, for me, represents the next big step in intelligence research. Every level of explanation for intelligence is a nightmare of complexity. But what else would you like to spend your time doing rather than untangle this complexity? This is really the challenge of our time. Emily Willoughby made a little, a very detailed illustration of the genes that had been identified in James Lee's first 1 million+ GWAS study. These are genes that have been identified as related to educational attainment. And she kind of laid out some of the things these genes do. So yes, it's going to be complex, but therein lies the challenge. As John F. Kennedy said in his famous Rice University speech, we choose to go to the moon not because it's easy, but because it is hard. And if you really want to be inspired about working on complex projects, listen to his speech. You can find it easily on the internet, and watch the HBO dramatic series From the Earth to the Moon. You will be inspired. And finally, I want to show you this small bottle of Southern Comfort. It was the tradition at Hopkins that when a graduate student finished their oral exams, they would go down the hall and wait in a room while the faculty decided whether or not they passed. And then the chairman would come down, give you the good news, and present you with this small bottle of Southern Comfort. I got this very bottle in 1975. And ever since, I have taken a small swig when I have had some academic achievement that meant something to me. It's my private celebration. I'm sharing it with you now. I just want to point out there's a small amount left, and I'm hoping to use it up. And so I'm going to stop there. I know it's been a very long day for many of you. It's the end of the day. Here in California, it's still the morning. It's the beginning of the day, and I'm looking forward to what happens next. So thank you very, very much.